Hello everyone, today we're going to talk about our special senses, gustation, olfaction, and auditory. Olfaction is the sense of smell, gustation is our sense of taste, and then we'll talk about our hearing ability and our equilibrium sensation. And we're going to discuss the anatomy. So we'll discuss the anatomy of our taste buds, our olfactory receptors, the anatomy of our ear. So let's go ahead and get started. Our sense of taste is known as gustation, and the sensory organs that would help us taste are known as our taste buds. Those taste buds have cells within them known as gustatory cells, and those are going to be our taste receptors. So those are the ones that are going to receive a stimulus, which is basically a molecule you are trying to taste, and they will fire off. They will send an electrical message down the neurons, and eventually the electrical message is going to go to the gustatory cortex in your brain so that your brain would help you interpret what you've just tasted. Those taste buds are mostly located on your tongue, but we also have a few scattered on our soft palate. We also have a few taste buds on the inner surface of our cheeks. We've got some on our pharynx, which is your throat, and we also have some located on our epiglottis. If we take a look at an image of the tongue right here, we all know that our tongue is not really smooth. There are those projections on our tongue, making our tongue like a bumpy surface. Those bumps that you see on your tongue, those are not taste buds. Those bumps are known as papillae, and inside the papillae are the taste buds. So those bumps, we have a bunch of those bumps on our tongue, close to the tip of our tongue. The bumps there, we refer to it as the fungiform papillae. Then we have more bumps scattered over here, kind of close to the mid part of our tongue, and those are known as the filiform papillae. On the sides, we have the foliate papillae, and then near the back of our tongue, we have these papillae right here, known as the valate papillae. So within those papillae are the taste buds. Not all of them, not all of those papillae, by the way, have taste buds. For example, the filiform papillae have no taste buds within it. If we take one of those papillae and go ahead and open it up, you would find the taste buds, which are, again, the sensory organs for our taste. So let's go ahead and zoom in on one taste bud and see what it consists of. So here is one taste bud. We've zoomed in on it. It kind of looks like a rosebud. And this taste bud right here has two major cells. It has the gustatory cells and it has the basal cells. Near the end of that taste bud right here, there is that pore, that taste pore. So for us to taste, it's actually easier for us to pick up tasting molecules if they are bathed in saliva. So if your tongue is pretty dry, it would actually take you longer to pick up those chemicals that you are trying to taste versus if your tongue has saliva on it. So once those tasting molecules are dissolved or mixed in with the saliva, it will make its way through that taste pore. And this is where it's going to go ahead and stimulate the gustatory cells. So those gustatory cells are our receptor cells for our taste, and specifically the part that will get that will be triggered when it comes into contact with a tastant molecule are long projections coming out from the gustatory cells, and it kind of projects through that taste pore, known as the microvilli, or we refer to them as the gustatory hair cells. So those gustatory hair cells, when they get into contact with those tastant molecules, they will fire off an electrical message, and you notice there are nerves right here that are connected to your gustatory cells, so they will pick up the message, and the message is going to eventually go to the gustatory cortex within our brain, so that our brain would interpret what we've just tasted. Another type of cell we have within our taste buds that are pretty important are the cells back here known as the basal cells. And basal cells are basically stem cells that can regenerate or help um, regenerate our epithelial cells, or our um, gustatory cells. Now, um, it takes every 7 to 10 days. So every 7 to 10 days, our basal cells can actually make new gustatory cells, which tells you that those gustatory cells, they do get damaged pretty fast. So about every week, your basal cells, your stem cells will go ahead and regenerate those gustatory cells for you. Um, let's see. Anything else I want to mention? As we age, of course, things tend to get slower, and so as we age, it becomes harder for us to regenerate those cells. It just takes longer, and so therefore we start losing a sense, like, we start losing the sense of taste, especially you would find older individuals asking for more salt to be added to their food or sugar, because it would be harder for them to pick up those tastant molecules. The 5 taste sensations are sweet, so we are able to pick up sweet taste, salt, sour, bitter, and umami. Umami is that savory taste. So when you say something like, pretty savory, like taste of cheese, for example, it's tangy. The beef taste, that's what umami is. We are actually pretty sensitive to bitter and sour, so we pick up bitter and sour way faster compared to picking up, like, taste of something sweet. And we're usually faster in picking up the bitter taste and the sour taste, because usually when things go wrong, they're either they either turn sour or they turn bitter. So it's a way for your body to protect you against eating something that has gone bad. Now, the nerves that are going to actually connect to your tongue and eventually pick up the message so that it could go to the gustatory cortex within your brain, those would be the facial nerve and the glossopharyngeal nerve. So they will pick up the message and eventually send it to the brainstem, and from there the messages are going to be picked up by some other neurons until it reaches the gustatory cortex in your brain. 80% of taste is actually smell. That's why when we get sick and your nose gets all stuffy and inflamed and you can't smell, you would notice that if you can't smell anything, then you're not going to be able to taste either. Let's talk about olfaction, and olfaction is our sense of smell. Now, for us to pick up these odorant molecules, one more time, Those odorant molecules need to dissolve in kind of the fluids that are coating our nasal membrane and get mixed in with the mucus in our nasal cavity to make it easier for us to pick up those odorants. We humans, we are not as sensitive in picking up lots of smells compared to other animals, like to dogs, for example. They have a better um, sense of smell compared to us. For us to pick up an odorant molecule, those odorant molecules, they must be volatile, meaning that they must be in a gaseous state as they enter into our nasal cavity. As they enter into our nasal cavity, you can see that right here at the roof of the nasal cavity, this is where we have our olfactory epithelium. Those odorant molecules, if we go ahead and zoom in as they are entering into our nasal cavity, so we're going to go ahead and zoom in on this region right here, you would notice that there is a mucus layer, and this mucus layer actually help us pick up those odorant molecules. So when those odorant molecules dissolve and get mixed in with the mucus, it makes it easier for us to actually pick up those odorant molecules. These are right here within the olfactory epithelium. We have those olfactory receptor cells, and they are the ones that are going to be triggered when an odorant molecule attaches to them. The ends right here of those olfactory receptors, there are those hairs, and those are not actual hair. So this is not the hair that you see inside of your nose. Those olfactory hairs are going to actually increase the receptive area. They're the nerve endings of those olfactory receptors that we have. Once an odorant molecule triggers off those olfactory hairs, an electrical message is going to move down those olfactory receptors, where the electrical message is going to move down the axon, and the axons are actually going to pass through the ethmoid bone. So here is the ethmoid bone right there. And so it's going to pass through it, and it's going to synapse with some other neurons, and they're going to synapse within the olfactory bulb. And the message is going to go down the olfactory tract, and from there it's going to go to the olfactory cortex in our brain, so that our brain would pick up the message. Now, those olfactory sensory neurons, they don't last forever. Their lifespan is from 30 to 60 days, and then we go ahead and replace them, because there are a bunch of basal cells. You see those basal cells right here, and those basal cells are basically stem cells. So those stem cells can actually regenerate those olfactory neurons every 30 to 60 days. As we age, this process becomes much slower, and so therefore, as we age, it becomes harder for us to pick up and smell those odorants as before. Our ears contain receptors that would help us with equilibrium, which is basically our balance. And our ears also contain some organs and some receptors that would help us hear. The ear is divided into three regions. We've got our external ear, our middle ear, and our inner ear. Our ear is divided, as we said, into three parts: the external ear, which is going to be this region right here, the middle ear, which is going to be this region right here, and then the internal part of our ear. The external part of our ear, it's made up of the auricle and the external acoustic meatus. The auricle is this outside part, and this actually auricle is mostly made out of elastic cartilage. It helps funnel the sound waves into your auditory canal. Your auditory canal is known as the external acoustic meatus. The external acoustic meatus, it's lined with hair and sebaceous glands that can produce cerumen, which is wax. The earwax is pretty important because it helps trap any foreign bodies entering into your ear canal. It also prevents insects from taking your ear canal as home. When sound waves enter into your external acoustic meatus, those waves are going to hit your tympanic membrane. And your tympanic membrane is basically your eardrum. When your tympanic membrane vibrates, the vibration of the sound is going to enter into your middle ear. But what is your tympanic membrane made out of? Your tympanic membrane is made out of a thin connective tissue membrane, and it's covered by skin. So as we said, when the sound waves hit your tympanic membrane, it's going to vibrate, and the vibrations are going to move into your middle ear. This is where you have the three smallest bones in your whole entire body, and those bones are known as your auditory ossicles. We've got three of them. The one that's connected and attached to your tympanic membrane, it's the malleus. Then comes the incus, then comes the stapes. Those are three small bones that are going to start vibrating when the sound vibrations vibrate the tympanic membrane. The first one that will vibrate would be the malleus, then the incus, and then the stapes. If those three ossicles do not vibrate, that can cause problems and it can cause deafness. We are going to zoom in later on those three bones right here and talk a bit more about them. But next comes your internal ear. Let's just kind of take a look at what your internal ear looks like. So the internal ear, we've got this part right here known as the cochlea. And actually the term cochlea means the snail. And it does look like a snail. And then we've got this part right here known as the vestibule, and we have those three semicircular canals. So we've got the cochlea, we've got this middle part, the vestibule, and the semicircular canal, and those are what we have in our internal ear. Connected to your internal parts of your ear, we have our vestibulocochlear nerve, and the vestibulocochlear nerve is going to send the message to your to the cortex in your brain, so that your brain, your auditory cortex, so that your brain would interpret what you've just heard. So one more time, the vibrations, the sound vibrations, the sound waves would enter into your external acoustic meatus, vibrating your tympanic membrane. The tympanic membrane, when it vibrates, those vibrations are going to move into your middle ear, vibrating the three ossicle bones, and then the vibrations are going to move into your internal ear. Within our cochlea, we have our sensory organs for hearing. The vibrations are going to trigger the little hairs, little nerve endings in your cochlea, sending a message down your vestibulocochlear nerve, and from there the message goes to your auditory cortex in your brain. There is this canal right here, which is known as your auditory tube, and your auditory tube, or we refer to it as your pharyngo-tympanic tube, it links your middle ear. So right here, it links your middle ear with your nasopharynx, which is the superior part of your throat. So it connects your middle ear cavity with your superior part of your throat, known as the nasopharynx. And there's a mucosa lining your middle ear, and it's the same mucosa lining that also lines that auditory tube. And so that's why, because your middle ear is connected with your throat, sometimes what happens when you are sick and you get a sore throat, the infection can move from your throat into your ear. It happens mostly with kids because their pharyngotympanic tube is much shorter and more horizontal looking. Now, our pharyngotympanic tube, as like in adults, it's flattened and it's closed, but when you swallow and you yawn, it opens up so that it can equalize the pressure basically in your middle ear with the pressure of the external air. And you must have experienced that equalizing of the pressure between the air and your middle ear. If you ever yawned and it felt like your ear was popping, that ear popping sensation, that means that the pressure is equalizing in your middle ear with the outside air. With outside air pressure. So usually when you are on a plane and the pressure up there starts changing and you start feeling that you can't hear that well, and the sound is muffled and you feel pressure in your ears, they would tell you either yawn or open and close your mouth or start chewing gum, because that's going to help equalize the pressure in your middle ear. If we zoom in on that middle part of our ear, which consists of our three ossicle bones right here, and then right here would be the auditory tube or the pharyngotympanic tube. As I said, that would help equalize the pressure in your middle ear with the air pressure on the outside. Those three bones, as I said, need to vibrate so that the vibrations would get transmitted into your internal ear so that we could hear. Those three bones right here, the malleus, the incus, and the stapes, you would notice that there are actually two muscles that are connected to these middle ear bones right there. And what are those muscles for? So in case you get exposed to a very loud sound, those muscles would actually contract and they would limit your ossicles from vibrating, minimizing the damage that can happen to your hearing receptors. This image right here, it's a zoomed in image of what we have in our internal ear. As we said, we've got the cochlea, we've got the vestibule, and we've got those semicircular canal. Our internal ear looks like a big maze, as you can see, and so we refer to it as the labyrinth, which means maze. There is the bony labyrinth, which is those regions right here, kind of like that outside margin. We refer to it as the bony labyrinth, and it's those channels worming through. The bony labyrinth, it's filled with a fluid known as perilymph. So right here, that fluid known as perilymph is basically filling up all of these regions right here, filling up your bony labyrinth. Suspended in that fluid, suspended in that perilymph fluid, there is the membranous labyrinth, which you can see right here depicted in the color pink. So throughout your entire bony labyrinth, there are those series of membranous sacs and ducts, which we refer to as the membranous labyrinth. And inside of the membranous labyrinth, there is a fluid known as the endolymph. Those two fluids, the perilymph and the endolymph, those two fluids are going to help conduct the sound vibrations which are involved in hearing, so that we are, eventually those sound vibrations would trigger your vestibulocochlear nerve, and the message would get sent to your brain for us to interpret what we are hearing. those fluids are also actually going to respond to changes in your body position. That's why we say that your ears can help us detect our body positions, and they can also help us detect your body movement, and whether you're running, whether you're sitting down, and it also helps you with your balance. And so that's why you would notice when someone like spins really fast, they would start feeling dizzy because the fluids in your ear, especially in your internal part of your ear, are transmitting those messages, and eventually you will feel dizzy. Those semicircular canals right here that you see, those semicircular canals are actually containing some receptors that would respond to the rotational movement of your head. So if you rotate your head, your semicircular canals would actually be able to pick up those movements and send a message so that, again, your brain would interpret that you are moving your head. The cochlea, which is that snail part right here, it houses the receptor organs of hearing, known as the organ of Corti. And those receptors, again, when the sound waves make its way deep into your internal ear, those receptors would get triggered, and eventually the message is going to go down your vestibulocochlear nerve. To be more precise, it would be the cochlear branch, because the vestibulocochlear nerve branches into the cochlear branch and the vestibular branch.